Phoenician origins, DNA, genetics. Ancient mitochondrial DNA of Phoenicians from Sardinia and Lebanon, a story of settlement, integration, and female mobility. Adapted from a PLOS.org, open archive article and narrated by D.W. Draffin. The aim of this study was to seek mitochondrial DNA markers recovered from ancient samples that can be associated with Phoenician origins and ancestry, enabling us to track Phoenician mobility and genetic impacts of settlement, in this case in Sardinia. We collected ancient samples from documented Middle Bronze Age and Phoenician burials in Lebanon and from Monte Sirai, Sardinia, one of the largest Phoenician Punic necropoli in the central Mediterranean. The Phoenicians, one of the great ancient civilizations, emerged in the northern Levant around 1800 BCE, and by the 9th century BCE had brought together East and West by spreading their culture across the Mediterranean basin. They linked Asia, Europe, and Africa through their trade networks and settlements, and they created the ancestral alphabet of most of the Western world today. They were skilled navigators whose trade networks extended throughout the entire Mediterranean basin, and they had completed an Egyptian-sponsored circumnavigation of Africa long before the Vikings ventured out of the sight of land. Despite their widespread influence, what is known of the Phoenicians comes from what was written about them by the Greeks and Egyptians. In this study, we investigate the extent of Phoenician integration with the Sardinian communities they settled. We present 14 new, ancient mitogenome sequences from pre-Phoenician, circa 1800 BCE, and Phoenician, circa 700 to 400 BCE, samples, 4 from Lebanon and 10 from Sardinia. We then compare these with 87 new, complete mitogenomes from modern Lebanese and 21 recently published pre-Phoenician ancient mitogenomes from Sardinia to investigate the population dynamics of the Phoenician Punic site of Monte Sirai in southern Sardinia. Our results indicate evidence of continuity of some lineages from pre-Phoenician populations, suggesting integration of indigenous Sardinians in the Monte Sirai Phoenician community. We also find evidence of the arrival of new, unique mitochondrial lineages, indicating the movement of women from sites in the Near East or North Africa to Sardinia, but also possibly from non-Mediterranean populations and the likely movement of women from Europe to Phoenician sites in Lebanon. Combined, this evidence suggests female mobility and genetic diversity in Phoenician communities, reflecting the inclusive and multicultural nature of Phoenician society. The details. From their homeland in what is today Lebanon, the Phoenicians sailed extensively across the Mediterranean for trade and established settlements in Cyprus, Malta, Sicily, Sardinia, Ibiza, the Iberian Peninsula, and along the North African coast, most notably Carthage. Their naval dominance was respected throughout the Mediterranean, and they provided maritime support to the Persians and the Egyptians. Despite their enduring influence, scant historical documentation attributed directly to the Phoenicians exists. Most of the Phoenician historic documents were written on papyrus and have not survived or been discovered yet. What we do know of them is what others, the Greeks, Romans, Egyptians, wrote about them. They were never a unified political state or distinct ethnicity within their homeland. The emerging coastal city-states of Byblos and Sidon thrived as maritime trade centers during the 3rd and 2nd millennia BCE, and later the city of Tyre. During the 1st millennium BCE, their occupants were referred to by the Greeks, and thus today, as the Phoenicians, from the Greek Phoinike, or Purple Country. in reference to their production of the valuable purple dye used in textile production. Prior to this, however, the region was occupied by Canaanites, who, due to political events in the north, south, and east, were confined to the thin coastal strip between the steep, cedar-covered mountains of Lebanon and the eastern Mediterranean. This coastal isolation meant that Phoenicians could only expand westwards, and this they did, targeting sources of valuable metals including silver and tin. They established settlements across the Mediterranean and dominated maritime trade networks for centuries. Starting in the middle of the 8th century BCE, the Assyrians waged war on the Phoenicians, and soon after they brought them under their yoke. With the decline of Phoenician influence in the east, Carthage, a city that was settled by Phoenicians fleeing Tyre in 813 BCE, became the center of Western Phoenician or Punic dominance until its destruction in 146 BCE by the Romans in the Third Punic War. Carthage, like many of the early Phoenician cities around the Mediterranean, adopted many of the rituals and features of their homeland. The island of Sardinia, like Cyprus, Malta, Sicily, and Ibiza, was a significant location in the early Phoenician trade routes to the Iberian and North African coasts, and later in the Punic interaction sphere. Phoenician settlements were established primarily along the south and west coast of Sardinia, with two of the earliest located at Sulcis and slightly inland Monte Sirai. As was common in Phoenician Sardinia, Monte Sirai was built next to the previous Nuragic tower used by the first inhabitants like a shrine. The necropolis at Monte Sirai was one of the largest Punic burial sites in Sardinia and was used between the end of the 7th and the first half of the 4th century BCE. This period of use coincides with the Carthaginian dominion in Sardinia, starting at the end of the 6th century BCE. Genetic research on modern populations in Lebanon and the Levant, the homeland of the Phoenicians, shows that much of the genetic structure of the region today is influenced by historic migrations associated with the spread of major religions, Christians, Muslims, and Druze. Research on the Y chromosome DNA of men currently living in locations of historic Phoenician influence from across the Mediterranean, compared with men from nearby locations with no evidence of Phoenician contact, has identified likely Phoenician Y chromosome markers. These markers were present in more than 6% of the men living in Phoenician-influenced locations around the Mediterranean, and in more than 30% of Lebanese men. Comparative analyses of uniparental genetic markers in modern Lebanese indicate different genetic histories between males and females, with significantly more European lineages evident in the maternally inherited mitochondrial DNA than observed for the paternally inherited Y chromosome. When these European-derived lineages were integrated into the Lebanese population is not yet fully resolved. If identified, mitochondrial markers associated with Phoenician settlement around the Mediterranean may help document the movement of women within the Phoenician networks. They can also be used to indicate how much integration with indigenous inhabitants occurred in Phoenician settlements. We now have the opportunity to investigate the genetic and cultural history of the Phoenicians and their spread across the Mediterranean through the analysis of ancient DNA from skeletal remains recovered from pre-Phoenician, Phoenician, and Punic archaeological sites across the Mediterranean. A total of 28 ancient tooth samples were obtained for ancient DNA analyses from 4 archaeological sites in Lebanon, including Tel Fadus Kfar Abida and one site, Monte Sirai, in Sardinia. The archaeological context of the samples from the first Lebanese site relates to the late Phoenician Achaemenid period, 539 to 330 BCE. In addition to the stratigraphy, a pilgrim flask, a dish, a pitcher, a gold ring, and red carnelian beads were found in the tomb, enabling accurate dating of this archaeological context. At one site in Beirut, the skeletal remains of children were found in pits excavated in the geological substrate. All these burial rituals are consistent with a 5th to 4th century BCE date. At Tel Fadusk Farabida, a town situated 2 kilometers south of the coastal town of Batroun, we sourced our samples from 3 tombs. The pottery from the 3 tombs is quite homogeneous and can be dated to the end of the Middle Bronze Age I, circa 1800 BCE. This dating is also supported by a scarab found within one of the burials, which dates to the late 12th to 13th Egyptian Middle Kingdom dynasty. The archaeological site of Monte Serai is a hilltop settlement located in Carbonia, southwestern Sardinia, Italy. The site is comprised of 3 large sectors. The main sector of the settlement, the Acropolis, located in the southern portion of the hill, the Sacred Place, or Tophet, located in the northern portion of the site, and the large necropolis located in the valley separating the settlement from the Tophet. Archaeological evidence indicates that Monte Serai was founded by Phoenicians in the last quarter of the 8th century BCE and totally abandoned during the 1st century BCE. The necropolis contained several tomb types, including inhumations of single and multiple individuals, primary cremations, enkitrismoi, child burials in transport amphorae, and semi-combustions. The archaeological interpretation of the group burials in Monte Serai is that they could represent family burials. We found that one group burial, containing 4 individuals, included 2 6 to 12-year-olds who had near-identical haplotypes. They also shared 2 novel mutations. It is therefore possible that these 2 were indeed siblings or otherwise maternally related, for example, cousins. The other 2 burials in that group had 2 non-local lineages, another 6 to 12-year-old and a young adult female 16 to 22 years of age. While this indicates that there is not a maternal connection between these 4 individuals who were buried together, it is possible that they are family members related through paternal connections. Analyses of more burials from Monte Serai and possible extended analyses of Y chromosome or autosomal data are necessary to further test the relatedness of individuals buried in Monte Serai. Without ancient DNA, a Phoenician-introduced signature can be difficult to distinguish from earlier Near Eastern lineages that spread during the Neolithic expansion to European sites that were later colonized by the Phoenicians like Monte Serai. It is expected, however, that Neolithic expansions would spread lineages broadly across the landscape, where Phoenician signatures are likely to be restricted to locations of well-established Phoenician contact. Due to the relative isolation of Sardinia and the unusual genetic signature of its population, modern Sardinians are often presented as the best representation of the early Neolithic population that expanded from the Near East into southern Europe. While the later Phoenician influence in Sardinia was likely via Carthage, as indicated by the archaeological evidence from Monte Serai, we still might expect to see the appearance of new Near Eastern and/or North African mitochondrial lineages in the burials from this site, given that Carthage was settled directly from Tyre. The most common haplogroup seen in our ancient Lebanese and Sardinian samples was the superhaplogroup H, identified in 7 of 14 samples. 2 of the 4 ancient Lebanese samples belong to haplogroup H, and a third belongs to the sister clade R0. 5 of the Monte Serai samples, 50%, were identified as having H haplotypes. There were no shared haplotypes between the ancient samples of Lebanon and those from Monte Serai. However, All sequences belonging to haplogroups H and R0, ancient samples of Lebanon and Sardinia and Montserrat, are very closely related, as they are separated by only a few mutations. In modern populations, the frequencies of haplogroup H range between 40 to 50% in Europe and about 20% in Southwest Asia. Subclades H1 and H3, the major subclades of this haplogroup, are almost exclusively European. H1 and H3 are the most common found in modern Sardinians. No H3 lineages were found in our ancient or modern Lebanese samples. Its presence in Sardinia in ancient samples, both pre-Phoenician and Phoenician, combined with the diversity seen in the modern Sardinian population, is consistent with at least a Neolithic introduction to the island, if not pre-Neolithic. Haplogroup HV is also an ancient European lineage, likely originating in the Mediterranean region during the last glacial maximum, and has been identified in early Neolithic remains from Spain. Two samples from Montserrat have J1c haplotypes. This full motif was not found in any of our modern Lebanese samples. This haplotype was identified in one pre-Phoenician sample dating to around 4000 BCE in the Near East. It is clear that J1c was present in Sardinia prior to the arrival of Phoenicians, and possibly prior to Neolithic expansions. The presence of one sample from Montserrat, a 6 to 12-year-old child that has an N1b haplotype, is of particular interest. Haplogroup N lineages are rare in modern Sardinians. Brandt and others suggest that N is a marker of Western European hunter-gatherers, as it has been found in Mesolithic samples from Portugal and a Paleolithic sample in southern Italy. Ancient samples with haplogroup N1a have been found in early Neolithic sites from Spain and Germany, but N1b has not been recorded in Neolithic samples outside of the Levant, with two exceptions from Anatolia. N1b is a relatively common haplogroup in Lebanon, occurring in 10% of modern samples. While we have not found any N1b in our ancient Lebanese samples, it is not unlikely that this haplogroup was introduced to Sardinia via Phoenician contact, either directly from the Levant or via Phoenician Punic settlements in North Africa, for example, Carthage. N1b-1a lineages have been identified in modern Tunisians and in a modern Moroccan, as well as in a modern individual from Sicily, another island with known Phoenician settlement. Haplogroup W is thought to have evolved during the last glacial maximum around the Caspian Sea region. Haplogroups W1, W3, and W6 have been found in late Neolithic and early Bronze Age sites in Germany and Russia, and W1 has been identified in a Neolithic sample from the Iberian Peninsula. While haplogroups W1, W3, and W10 are present in modern Sardinians, the archaeological sample from Montserrat of a young female included in a group burial is, as far as we know, the first W5 identified in Sardinia. Our ancient result from Montserrat is indeed significant and establishes a minimum date of late 5th century BCE for haplogroup W5 in the Mediterranean region and, given the Phoenician trade networks, could explain the presence of W5 in North Africa. It is possible that haplogroup W5 got into the Mediterranean via the Phoenician tin trade with Britain or Ireland, though only further ancient DNA work can confirm this hypothesis. A sample from Montserrat belonged to haplogroup X2b. Haplogroup X is relatively rare in Europe, generally found at frequencies of less than 1%. The highest frequencies of X in Europe are reported in Catalonia, the Pyrenees, and southern Portugal, at about 2.5%. It is found at relatively high frequencies in Druze from the Levant, where it reaches frequencies of up to 15%, though we did not identify X haplotypes in any of our modern Lebanese samples. It is possible that X2b was a Phoenician haplogroup introduced to Sardinia either directly from Lebanon or via North Africa, though an early Neolithic introduction, perhaps via a maritime route, cannot be rejected. However, haplogroup X was not found in any of the 21 ancient Sardinian samples of earlier studies. And finally, a sample in Beirut was identified as a T2b, a haplogroup that we did not identify in any of the modern Lebanese we sequenced, though T2 is found at low frequencies there today and was identified in one of our modern Lebanese samples. Haplogroup T2 is currently found at around 10% frequency in northern and central Europe, though some of the highest rates are in Sardinia, southern Portugal, and pockets in northern Spain. T2b is the most common T2 subgroup found in Europe today, and it has been identified in ancient samples from Central Europe. There has been some debate regarding the origins of the T2 lineages, which were originally thought to have had a Near East origin and spread into Europe with Neolithic expansions. It is possible that the origins of T2, and in particular T2b, may indeed be in southern Central Europe in the last glacial maximum. The presence of a T2b3 haplotype in an ancient sample from Lebanon is unusual, as it does not appear to be an indigenous lineage. The tomb in which the burial was found was not marked, and did not have any of the artifacts found in other sites, suggesting different burial customs. This is further evidence that this individual belonged to a different socioeconomic class or ethnic group, perhaps a slave brought to Beirut from one of the Phoenician settlements, such as Cagliari, Sardinia, formerly Carali. Our ancient Beirut sample is most closely related to modern samples from Tunisia, again, possibly indicating movement of women through the Phoenician networks from European colonies and trading ports to Phoenician cities in the homeland and Carthage. Earlier studies suggest that Sardinian-specific haplogroups and ancient mitogenomes dating to between 6,000 and 3,000 years ago may indicate that the earliest inhabitants of the island arrived prior to the Neolithic expansion into the region, which is consistent with archaeological and Y chromosome data suggesting earlier settlement. Recent analyses of 3,514 whole genome sequences of modern Sardinians suggest pre-Neolithic occupation of the island, and that some of the populations, particularly those in the mountainous central eastern Gennargentu region of the island, carry higher levels of this hunter-gatherer DNA compared to populations from more accessible regions. Our result may be indicative of early social and or geographic isolation of those hunter-gatherer populations. The proposed Phoenician lineages W5, N1B1A5, and X2b are not found in Sardinians today, suggesting rare introductions, of the nature of single individuals, rather than as representative of major colonizing groups. We suggest that N1B1A5 could be an introduction from Carthage or some other North African location within the Phoenician network. Conclusions. Our analyses of ancient pre-Phoenician and Phoenician mitogenomes from Lebanon and Sardinia provide important clues on cultural expansion, assimilation, and population mobility in the Mediterranean between the 5th and 3rd centuries BCE. First, we see a certain degree of continuity of population ancestry between Phoenician and pre-Phoenician populations in Sardinia, which is consistent with archaeological evidence of integration between the cultures. However, our data from Monte Sirai, combined with our previously published result identifying a European mitochondrial haplogroup U5B2C1 in a young man buried in a Phoenician crypt in Carthage, North Africa, provide evidence of several instances of unexpected, non-indigenous mitochondrial haplotypes in Phoenician burials, both in and outside the homeland of Lebanon. These include the T2b3 haplotype in Beirut, and the Near Eastern N1b1a5 and a Northern European W5, found in Monte Sirai, Sardinia. The Sardinian population is often described as the best representation of early farmer ancestry, and indeed, we do see the likely early farmer mitochondrial DNA lineages combined with those of the southwestern central Mediterranean Mesolithic, particularly haplogroups H, and possibly H1, H3, and H5 in the Phoenician samples at Monte Sirai. N1b1a5 and W5, which have not yet been recorded in modern Sardinian populations, and possibly X2b, we suggest, are examples of Phoenician introductions to Sardinia, not only from the Phoenician homeland or via Punic settlements in North Africa, but may represent the mobility of women through the wider Phoenician trade networks. While previous Y chromosome analyses of modern populations around the Mediterranean demonstrated the impact of Phoenician males on the genetic makeup of many communities, we now see that it is likely that the Phoenician trade networks or settlement strategies also included the translocation of women throughout the region, as well as the assimilation of indigenous women in Phoenician settlements. Analyses of genetic variation in modern Lebanese populations suggest closer affiliations with Europe based on mitochondrial lineages. Our result, identifying the European lineage T2b3 in the Beirut site, suggests that some of those European mitochondrial DNA lineages could be associated with the integration of European women in Phoenician communities in Lebanon. Finally, numerous studies published in the last 5 years highlight the importance of ancestral DNA studies in understanding population change through time, though most of these have focused on the Neolithic transition. While these studies tell a story of significant population replacement in Europe with the arrival of farmers, analyses of later historic burials associated with Roman Britain, for example, and now our data from Phoenician sites, demonstrate that both migration and cultural assimilation were common, resulting in surprisingly cosmopolitan communities in the past. My name is D.W. Draffin. I do voiceover, and I'm available for hire.